We're going to talk about genome engineering applications, and we're going to focus on ones that are less about the kind of overexpression or deletion that you would do to increase production of a particular product. And so takeaways for this lecture are that there are reasons to modify a genome besides knockouts and up or down regulation. So a big trend within metabolic engineering, and it's only naturally fitting when the field gets to a certain stage, is that you can readily, as many companies and academic labs have done, you can overproduce some metabolite. You can make so much of it, in fact, that you have a different problem. Your cells are struggling because of the product toxicity. Or, for your overall bioprocess, you might actually want the fermentation to happen at a different temperature, maybe to help with separation of the product or based on some other organism. And so there might be more complex phenotypes that you want to engineer, and including to product tolerance. And so that might require many different kinds of interventions on the genome. And the other trend that we're seeing is that you might want to make a lot of chemicals that aren't naturally made, meaning that aren't made by nature. And so those type of chemicals might not work with the cell's existing genomic framework. And we'll dive into one example from my graduate work there. Then there's so much work in the area of genome engineering geared towards different applications that we can talk about genomes that can be a lot smaller, that can be larger, that can be hybrids of different species, and that can just be quite radically different than our normal genomes. And so we'll spend some time talking about why people might want to do that. And what a good chunk of that will relate to is this idea that the future of protein engineering is going to be shaped by creative modifications to the genetic code. Because one of the real limitations for what you can do as a protein engineer is that you've got just your natural 20 amino acids that are encoded by codons in the codon table that are therefore in your genome. And so when you make changes to what codons are in your genome and you make associated changes with translation machinery, you can introduce new building blocks into proteins. And that may well be a way to get not just enzymes that give you more metabolic product, but also some more powerful therapeutics and other kinds of proteins. So a starting point, still thinking about metabolic engineering, is that example of product overproduction. And so many of the products in metabolic engineering are carboxylic acids. And when you overproduce simple organic acids of various chain lengths and aromaticity or aliphaticity, you get a lowering of pH because the carboxylic acid functional group is acidic. And when you've made so much of those molecules, you have a number of different kinds of problems that you can encounter related to stress of them being within the cell, and then the associated proton import or export. You've got cascades of stress response that could be activated. You may want to just for both separation, maybe during your fermentation, as well as to improve tolerance, you may want to secrete these products. Some of your kinds of products could destabilize your membrane. And so what both the figure on the left and on the right from two different papers kind of illustrate are examples where you might want to do some broader scale host or genome engineering to deal with these kinds of problems that all relate to having a particular type of product that you're trying to make that could be deemed as toxic to the cell, either once you've overproduced it, or maybe when you have very little of it, even to begin with. So the very little of it example is one where I'm going to spend more time. It's something that I'm quite familiar with, and I'll spend really more time diving into it on Thursday, just because it's almost noon already. But something that I just want to introduce this concept with is this idea that there's lots of different kinds of functional groups that organic chemists and we as consumers use and benefit from in our materials, in other kinds of products that are derived from petroleum. And genome engineering can help enable some of this unusual chemistry. And it's not exactly obvious why, so that's part of the reason why I'll go through this example. But suffice to say that here are 21 different functional groups found that in organic chemistry that are commonly used. And the ones in this sort of gold box are what I call biochemically restricted and common microbes, which means one of two things, or it means both of these two things. It means that if you provide these kind of molecules to a cell, and we'll focus on the aldehyde example, your cell, because of the genome that it has and many enzymes that it has, will very quickly, naturally take this aldehyde and convert it to something else, most commonly an alcohol. And so that can be a problem, right? That your cell has all these competing processes. Or it has, maybe a stress response gene that's just going to flare up because something looks like it's stressful, even though it might not actually be. And remember, all it has to do is potentially bind with a transcription factor that has to do with stress response. And it may not actually be, creating any other kinds of negative effects within your cell. So that's one problem, is that you've got something in your genome that makes this, imposes this kind of biochemical limitation or restriction. Another is that you just don't have enzymes known or in most microbes that can make these groups to begin with. And so some of these are present. Again, maybe they do show up in some places in nature, but not commonly, like alkanes. There are a number of cyanobacterial strains that can make these in very specific kinds of alkanes of very specific chain lengths. If we start thinking about, nitriles or isocyanates or azo compounds, a lot of the literature in the last 5 years has started to discover more and more examples of enzymes that can either do this in settings outside of the cell or when prompted to do this, but they don't naturally do this. Or increasingly, just in the last couple of years, there are some enzymes in very rare secondary metabolite biosynthesis pathways that are known to do this. So, the approach that, for example, my research group takes is we are very much under the belief that nature and living cells and systems can be engineered to do a lot more of this chemistry, and to be able to then really mimic more of the kind of products that that we would get from traditional petroleum, rather than relying just on what nature has already made somewhere else. But rather than tell you a story that's really geared towards unnatural molecules, where I'm going to pick up next time is on the idea of aldehydes, which do occur naturally, and in some organisms are retained much more efficiently than others. But in microbes, particularly model microbes like E. coli, these aldehydes are really unstable.